Ladies and gentlemen, very good morning to you all. We warmly welcome you all to the 12th International Conference on Dry Zone Agriculture, ICDA 2026, proudly organized by the Faculty of Agriculture, University of Jaffna, Sri Lanka, under the theme Smart Solutions for Commercial Agriculture.
Dear attendees, kindly complete your registration and please take your seat, as we are ready to commence the inaugural session of ICDA 2026. At this remarkable occasion, we extend our gratitude to the sponsors for their generous support, which has made this event a reality. Our platinum sponsor, JICA Presida, University of Jaffna; gold sponsor, National Science Foundation, Sri Lanka; bronze sponsors, Avon Palmo Chem Private Limited and World Vision; and other sponsors, Mr. Ponnambalam Suryakumar from USA, Northern Modern Irrigation, and Hatter National Bank. We are truly grateful for their invaluable contributions and proud to have their partnership in this meaningful occasion.
As we mark this special event, on behalf of the ICDA Organizing Committee 2026, we are honored to extend a warm and respectful welcome to our chief guest, Senior Professor Thirunavukarasu Velnambi, Vice Chancellor, University of Jaffna; Professor K. Bahirathan, Conference Chair and Dean, Faculty of Agriculture, University of Jaffna; our plenary speaker, Professor Ivan Oresnik, University of Manitoba, Canada; and keynote speaker, Professor Toshiaki Owari, the University of Tokyo, Japan.
We warmly invite the audience to join us in welcoming them.
Ladies and gentlemen, we now move to the auspicious moments of today's occasion, the lighting of the traditional oil lamp, a symbol of divine blessings, wisdom, prosperity, and the dispelling of darkness. We warmly invite everyone present to witness this graceful tradition as we seek blessings for the success of the conference and all its endeavors.
It's a great honor to invite our plenary speaker, Professor Ivan Oresnik from the University of Manitoba, Canada, to light the ceremonial oil lamp.
One to remain standing for the national anthem. Thank you, everyone. So please kindly take your seats.
Ladies and gentlemen, we now have a delightful performance for you. We warmly invite the talented team from the Sir Ponnambalam Ramanathan Faculty of Performing and Visual Arts, University of Jaffna, to present the welcome dance. Sorry for the inconvenience. While we warmly welcome all of you to this significant occasion, it's the time to hear from an esteemed member of our academic community who will officially extend the welcome on behalf of the organizing committee. It's our privilege to invite the convener of this conference, Dr. S. Tachnis Khan, to deliver the welcome address. Dr. Tachnis Khan, the podium is yours.
Makers, industry representatives, development practitioners, and students to share the knowledge, exchange ideas, and explore innovative solutions for the challenges facing modern agriculture. The International Conference on Dry Zone Agriculture has become a flagship annual event of the Faculty of Agriculture over the years. So ICDA has served as a vibrant platform for the disseminating scientific knowledge, fostering interdisciplinary research that contributes to the agricultural development, particularly in the dry zone environment. Through this engagement, this conference continues to play a significant role in advancing sustainable and resilient agricultural system both nationally and globally. The theme of ICDA 2026, Smart Solution for Commercial Agriculture, is timely needed and relevant in the present context. Agriculture today faces unprecedented challenges, including climate change, water scarcity, labor shortage, increasing production cost, market uncertainty, and growing demand for safe and nutritious food. To address these challenges, the agricultural sector must embrace innovation and technology-driven approaches that enhance productivity, efficiency, sustainability, and profitability. So a smart solution in agriculture encompasses a wide range of advancement, including precision farming, digital technologies, artificial intelligence, remote sensing, climate smart practices, data-driven decision making, sustainable resource management, and innovative value chain development. These approaches have the potential to transform traditional farming system into more resilient and commercially viable enterprise while ensuring environmental sustainability and food security. Through the presentation and the discussion at this conference, we hope to explore how science and technology and innovation can be effectively integrated into an agricultural system to meet these needs of the farmers, industry, and the society. So with this brief introduction, now I have the pleasant duty of extending my warm welcome to our distinguished guest. So first and foremost, on behalf of the organizing committee, I am deeply honored to welcome our chief guest, Senior Professor Tirunavakarasu Velnambi, Vice Chancellor of the University of Jaffna. Sir, we are privileged to your online presence and grateful for your continued support and guidance. I extend my sincere welcome to our plenary speaker, Professor Ivan Oresnik from University of Manitoba, Canada, and our keynote speaker, Professor Toshiaki Owari from University of Tokyo, Japan. So we are greatly honored to have internationally renowned scholars among us, and we eagerly look forward to hear your insightful presentation and valuable contribution to this conference. And I also wish to extend a cordial welcome to our former Vice Chancellors, Deans of the Faculties, JICA expert and members, Council members, provincial directors, deputy directors representing various institutions, distinguished invitees, academics from University of Jaffna and other universities, session chairperson, and the registrar, librarian, deputy registrar of the Kilinochchi premises, academic support staff, and students. Your presence adds immense value to this gathering. A special welcome is extended to all the presenters and participants who have traveled from near and far to this to share their research finding, experience, and expertise. Your contribution form of the foundation of this conference and help create meaningful discussion that advance agricultural knowledge and practice. I would also like to express our sincere appreciation and warm welcome to our sponsors and partners. So your generous support and the commitment have played a vital role in making ICDA 2026 in a reality. So as we commence this conference, I encourage all the participants to actively engage in the technical session, exchange the ideas with colleagues, and establish new collaboration, and take full advantage of these opportunities this conference provides. Together, through the scientific inquiry, innovation, and partnership, we can contribute towards building a more sustainable, resilient, and commercially successful agriculture sector for the future generation. So once again, I warmly welcome you all to the 12th International Conference on Dry Zone Agriculture, ICDA 2026. Thank you for joining us, and I wish you a productive and inspiring, memorable conference. Thank you very much.
Thank you, sir, for your inspiring words. At this moment, we proudly recognize the generous sponsors who have made ICDA 2026 possible through their unwavering support. Our platinum sponsor, Jacob Presida, University of Jaffna. Gold sponsor, National Science Foundation, Sri Lanka. Brown sponsors, Avon Pharmachem Private Limited and World Vision. Other sponsors, Mr. Ponnambalam Suryakumar from USA, Northern Modern Irrigation, and Hatton National Bank. We extend our sincere thanks to each of them for their valuable support.
Now, I request Professor K. Pakirathan, Conference Chair, ICDA 2026, and Dean, Faculty of Agriculture, University of Jaffna, to deliver his address.
Very good morning, ladies and gentlemen. I warmly welcome the Chief Guest and the Vice Chancellor, Chair Professor T. Velnambi, who joined this conference remotely due to his busy schedule in a Colombo meeting. And the plenary speaker of ICDA 2026, Professor Ivan Orsinik from University of Manitoba, Canada. And the keynote speaker, Professor Toshiaki Owari from University of Tokyo, Japan. Former Vice Chancellors, the Deans of the Faculties, and Council members, Directors, and Deputy Directors, Additional Directors of Department of Agriculture, Animal Husbandry, Irrigation, and Animal Production and Health, Heads of Departments, Professors, Senior Lecturers, Senior Assistant Registrars, Kilinochchi Premises, and the Works Engineer, Farm Manager, academic supporting staff, and my dear students. Gathered here to glorify this ICDA 2026. It's been organized by the Faculty of Agriculture with the theme of Smart Solutions for Commercial Agriculture. It is my great pleasure to deliver this conference chair address as the Dean of the Faculty of Agriculture in the 12th ICDA 2026. You all know that the ICDA is a unique conference specially designed to address the problems in the dry zone agriculture. This concept of ICDA incepted in 2015 at the celebration of the Silver Jubilee year of this agriculture 2015. This ICDA from 2015 is steadily growing and continuously organizing this annual event, and it's fixed as an annual event of this Faculty of Agriculture's annual calendar. Due to its continuous organization, it's reached the global community of the professional agriculturists. That's what every year from different part of the world, they are sending papers because they recognize the ICDA is a unique conference to address the problem of this agriculture in the dry zone agriculture. Today, we are going to get more information from speakers, but I have to highlight two key points, the alarming highlights. You all know that everywhere we are struggling to produce food because every year the increasing population is demanding to produce food. It is estimated to reach the 10 billion 2050. As per the current production, it is estimated that the FAO is saying annually we should increase the annual production by 1.7% to meet the demand of global population. But the agricultural scientists and farmers are facing problems, abiotic stresses and abiotic stresses. Can you imagine how we can achieve this global demand of food? Because the land is deteriorating, there is no more land for the cultivation, how we can increase the productivity? The second thing is the climate dilemma. You know that after 1877, we are going to face another super El Nino because of increasing the temperature in 2 to 3 degrees Celsius in the Pacific Ocean. It is going to create the mess in the all around the global, particularly the South Asian island nations like Sri Lanka. If the temperature is increasing, it will create the problem of, because we are receiving the rainfall in Jala and Maha season, it's going to shift. As per the schedule, the farmers cannot start the cultivation. Even the increasing the temperature, the production of the crops is going to lose its pollen viability. Deterioration of the land. We are going to lose the productivity because of the pollen, but their plants will not produce more. Due to the drought, severe drought we are expecting. What is the solution? That's how this in the ICDA from 2015, we are forecasting the theme, because you all know that in the dry zone, anytime, anything can happen. That's what we are focusing in this year theme, 2026, Smart Solutions for Commercial Agriculture. You know that without smart technology, incorporation is failed into the agriculture sector. At the current scenario, we cannot expect that expected outcome from the farmer fields. So therefore, this 12th ICDA conferences, we are focusing on how this globally reputed scientists, they have done the researches related to that smart agriculture principles, how it can be suited to this northern Sri Lanka, because in the northern Sri Lanka, we are facing problems after and after. You can see 30 years of the conflicts, we lost the production. We are in the last, northern province is the last. Again, COVID pandemic, supply chain and the production created a mesh. Then economic crisis. We are now suffering to get enough amount of food. We are begging the other countries. Again now the Strait of Hormuz closer, the shortage of fuel. Again the farmers and the productions are being affected. Now we are forecasting El Nino in 2026 to 2027. But northern Sri Lankans are revival community. In any type of hurdles, they can bring back to that and restore their cultivations and the food production. So therefore the scientists visited here because the two scientists from the two ends, one is from Canada, Manitoba, particularly his research focus is to soil microbiology, and another professor from the University of Tokyo from forestry. These research ideas is going to be addressed here and going to create a bigger network because the many agricultural academics, they have enjoyed the research flavor from Japan, Korea, Norway, Germany, India, UK, Canada, and US and Australia. So the bringing back these scientists and academic staff, now they have developed that their academic qualities by investing of this JICA grant aid project, by developing a very state-of-art research and training complex. Then Indian government invested on the many equipments and the capacity building. Again, JICA Presida is investing on capacity building and research and outreach. So therefore, today, these researchers present here will share their findings. These findings will give a clue what are the research gaps our students and academics going to address. They can snatch as a research idea. The ladies and gentlemen, you know that the ICDA is giving a wonderful opportunities for academics and the policymakers to share the ideas and developing the dry zones of Sri Lanka to increase the food production, as the present government is forecasting for the self-sufficient production without importation. So the success of the ICDA is not a simple task. Many nights, many brains, with their dedicated times, they tirelessly worked with the communicating with the university administration, the negotiation with the sponsoring partners. These all, now you all are witnessing how this ICDA 2026 is organized wonderfully. With this, I would like to conclude my Dean address or conference chair address. Thank you very much for the patience, and your continuous support in future is also expected. A different part of the world, the different reviewers, they screen abstracts submitted for the ICDA, more than 120 abstracts submitted, but we only accepted 62 abstracts to 63 abstracts because we have to maintain the quality. And also these accepted presentations also going to be evaluated by renowned evaluators came up from the different part of the Faculty of Agriculture. So with this message, I would like to conclude. Thank you very much for also once again. Thank you very much on behalf of Faculty of Agriculture and ICDA committee. Thank you very much.
Thank you, sir, for your insightful remarks. We are grateful for your support and valuable contribution to the success of ICDA 2026.
Ladies and gentlemen, we warmly invite the talented team from Sir Ponnambalam Ramanathan, Faculty of Performing and Visual Arts, University of Jaffna, to present the welcome dance.
गणपदे मग गणपदे नमो नमो मग गणपदे नमो नमो श्री मग गणपदे नमो नमो श्री मग गणपदे नमो नमो श्री मगेश्वर पुत्र मग गणपदे नमो नमो श्री मगेश्वर पुत्र श्री गणपदे मग गणपदे नमो नमो श्री मगेश्वर पुत्र श्री गणपदे मग गणपदे मग पंडित ज्ञान स्वरूप मग पंडित ज्ञान स्वरूप बादल दिलग गदंद बादल दिलग गदंद मग गणपदे नमो नमो श्री मगेश्वर पुत्र श्री गणपदे मग गणपदे मूलाधार क्षेत्र स्वरूप मूसिग वगन मोदक बालचंद्र कंद सोदरा ज्योतिमय काम कोडी कामाची कुमार मग गणपदे नमो नमो श्री मगेश्वर पुत्र श्री गणपदे मग गणपदे जनकिटतां दत्तितां तत्तितां द जनकिडतगतां जनकिडतां ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ಜನ್ನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ಜನ ಕಿಟ್ಟ ತಾಂ ತತ್ತಿ ತಾಂ ತತ್ತಿ ತಾಂ ತ ಜನಗಿಡ ತಗ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಾಂ ಜನಗಿಡ ಜನಗಿಡ ತಗ ತಾಂ 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 ತದಿ ತಾಂ ತದೀಂ ದೀಂ ದೀಂ ತಗತ್ತೋಂ ತದೋಂ ತೋಂ ತೋಂ ತಗತ್ತೋಂ ತನಂ ನಂ ನಂ ನಿಮಿತ್ತಾಂ ತದಾಂ ತಾಂ ತಾಂ ತದಿ ತಾಂ ತದೀಂ ದೀಂ ದೀಂ ತಗತ್ತೋಂ ತದೋಂ ತೋಂ ತೋಂ ತಗತ್ತೋಂ ತನಂ ನಂ ನಂ ತಗತ್ತಾಂ ತದಿ ತಾಂ ತಗತ್ತಾಂ ಜನು ತಾಂ ತದಿಮಿತ್ತಾಂ ತಾಂ ತರಿದ ತನದ ಜನುದ ತಿಮಿದ ತದಿಂಗಿಣದಂ 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 ತದಿತ್ತಾಂ ತಗತ್ತಾಂ ಜನು ತಾಂ ತದಿಮಿತ್ತಾಂ ತಂ ತರಿದ ತನದ ಜನುದ ತಿಮಿದ ತದಿಂಗಿಣದಂ 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 ತಜಂ 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 ದ ತನ 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 ತ ಜನು 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 ತ ತಿಮಿ 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 ತ ಜಂ ತೆರಿ ಗಿಣದಗ ಜಗದರಿ ಗಿಣದಗದಂ ಗಿಣದಗ ಜಗದರಿ ಗಿಣದಗದಂ ದಗದರಿ ಗಿಡದಗದಂ ಗಿಡದಗದಂ ತಿತ್ತಲಾಂಗಿದಗ ಜಂ ತೆರಿ ಗಿಣದಗ ಜಗದರಿ ಗಿಣದಗದಂ ಗಿಣದಗ ಜಗದರಿ ಗಿಡದಗದಂ ದಗದರಿ ಗಿಡದಗದಂ ಗಿಡದಗದಂ ತಿತ್ತಲಾಂಗಿದಗ ಜಂ ತೆರಿ ಗಿಡದಗ ಜಗದರಿ ಗಿಡದಗದಂ ಗಿಡದಗ ಜಗದರಿ ಗಿಡದಗದಂ ದಗದರಿ ಗಿಡದಗದಂ ಗಿಡದಗದಂ ತಿತ್ತಲಾಂಗುದಗದಂ ಗಣಪತೆ ಪ್ರಭೋ ಗಣಪತೆ பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே 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 பரிபூரண சார்ந்து வணங்கி துதி பாடியாடியுந்தன் சன்னதி சரணடைந்தோமே சார்ந்து வணங்கி துதி பாடியாடியுந்தன் சன்னதி சரணடைந்தோமே சார்ந்து வணங்கி துதி பாடியாடியுந்தன் சன்னதி சரணடைந்தோமே சார்ந்து வணங்கி பாடியாடியுந்தன் சன்னதி சரணடைந்தோமே சார்ந்து வணங்கி துதி பாடியாடியுந்தன் சன்னதி சரணடைந்தோமே சாந்த சித்த சௌபாகியங்கள் யாவையும் தந்தருள் சத்குருநீயே சாந்த சித்த சௌபாகியங்கள் யாவையும் தந்தருள் சத்குருநீயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பிரபோ கணபதே ஆதிமூலகணநாத கஜானன அற்புத தவல ஸ்வரூபா ஆதிமூலகணநாத கஜானன அற்புத தவல ஸ்வரூபா தேவதேவ ஜய விஜய விநாயக சின்மய பர சிவ தேவா தேவதேவ ஜய விஜய விநாயக சின்மய பர தேவ தேவா பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே தேடி தேடி எங்கோ ஓடுகின்றாருள்ளே தேடி கண்டு கொள்ளலாமே தேடி தேடி எங்கோ ஓடுகின்றாருள்ளே தேடி கண்டு கொள்ளலாமே கோடி கோடி மத யானைகள் பணி செய்ய குன்றென விளங்கும் பொம்மானே குன்றென விளங்கும் பொம்மானே கோடி மத யானைகள் பணி மத யானைகள் பணி செய்ய குன்றென விளங்கும் பொம்மானே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பரிபூரண வாழ்வருள்வாயே பிரபோ கணபதே பிரபோ கணபதே
Thank you for the elegant and mesmerizing dance performance, ladies. this remarkable occasion, we extend our gratitude to the sponsors for their generous support, which has made this event a reality. Our platinum sponsor, JICA Presida, University of Jaffna. Gold sponsor, National Science Foundation, Sri Lanka. Bronze sponsors, Avon Palmo Chem Private Limited and World Vision. And other sponsors, Mr. Ponnambalam Surya Kumar from USA, Northern Modern Irrigation and Hatter National Bank. I would now like to invite the following dignitaries to take their seats at the head table and grace us with their esteemed presence. Professor Ivan Orensnik, Plenary Speaker, University of Manitoba, Canada. Professor Toshiaki Owari, Keynote Speaker, the University of Tokyo, Japan. Professor K. Bakirathan, Conference Chair and Dean, Faculty of Agriculture, University of Jaffna. Dr. S. Takshaniskanth, Convener of ICDA 2026. Dr. S. J. Arasagesari, Finance Chair, ICDA 2026. And Dr. Mrs. A. Sayandan, Joint Secretary, ICDA 2026.
Ladies and gentlemen, as our distinguished guests now join us at the head table, we are delighted to formally begin the inaugural ceremony of ICDA 2026, a one-day research conference hosted in this vibrant and intellectually rich forum.
Ladies and gentlemen, the next much awaited item on our agenda is the official release of the proceedings of the 12th International Conference on Dry Zone Agriculture, ICDA 2026. Let us all witness this symbolic launch, which represents the collective effort, knowledge, and contributions of researchers from around the globe.
To officially launch the proceedings, we kindly invite the convener of this conference, Dr. Takshaniskanth, to officially issue the conference proceedings to our conference chair. Thank you, sir. We kindly request our conference chair to stay on the stage to issue the proceedings to the following invitees. We kindly request Professor Ivan Orensik, our esteemed plenary speaker, to receive the proceedings from our chief guest. Followed by Professor Toshiaki Owari, keynote speaker. I invite Professor T. Mitsunaka, Project Chief Advisor, JICA Presida, University of Jaffna. I invite Dr. Sasaki, Project Expert, JICA Presida, University of Jaffna.
we kindly invite Professor N. Pradeep Raja, Dean Arts, Acting Dean Graduate Studies. Professor N. Gangadharan, Dean, Management Studies and Commerce. Dr. A. Kaneshwaran, Dean, Engineering. Professor R. Surendra Kumaran, Dean, Medicine. And Dr. T. Kedison, Dean, Technology.
Also, we kindly invite Mr. S. Rajesh Khanna, Additional Director, Agricultural Research, RARDC. Dr. S. Gajarajan, Veterinary Surgeon. Dr. P. Gangadharan, Veterinary Surgeon. Dr. Mrs. S. Kirijagala, Deputy Director, Department of Animal Production and Health. Mrs. V. Sodhi Lakshmi, Deputy Provincial Director, Agricultural Extension. Ms. N. Bamitra, Assistant Director of Agricultural Research. Mrs. Tinarthiga, Assistant Director, Agricultural RARDC. Professor S. Sivajoganathan, Council Member. Professor P. Aingaran, Senate Representative. Professor S. Mohanadas, former Vice Chancellor. Professor P. Balasundaram Pillai, former Vice Chancellor. Dr. Mrs. Sahaya Mary, Department of Animal Production and Health. Mr. A. Amridalojan, Assistant Director. Ms. K. Patiraja, Senior Assistant Bursar, Kilinochi Premises. Mr. N. Selvanathan, Retired Research Officer.
May I respectfully invite one representative from each of our sponsoring organizations to kindly come forward to receive a copy of conference proceedings. Our platinum sponsor, JICA Presida, University of Jaffna. I kindly invite one representative from JICA Presida, Megumi. Our gold sponsor, National Science Foundation, Sri Lanka. Our bronze sponsor, Avon Palmo Chem Private Limited. Our bronze sponsor, World Vision. And other sponsor, Northern Modern Irrigation. And Hatter National Bank. Thank you, sir, for officially releasing the conference proceedings. Dear all, the conference proceedings are available digitally through the QR code printed on your name tag. Please feel free to send it and feel free to scan it and access the proceedings at your convenience. We respectfully request the dignitaries seated at the head table to take your seats in the front row. Ladies and gentlemen, the next highlight of our agenda is the plenary speech. May I now request Senior Professor Mrs. Nalina Gnanavil Raja to introduce our plenary speaker, Professor Ivan Oresnik from University of Manitoba, to our esteemed audience.
Good morning, distinguished guests, colleagues, and participants. It is my great honor to introduce our plenary speaker, Dr. Ivan J. Oresnik, a professor and a distinguished scholar in the field of molecular biology and plant-microbe interactions. Professor Ivan obtained a BSc in molecular biology and genetics in 1986, followed by an MSc in molecular biology in 1990 from the McMaster University, Canada. He read his PhD in plant physiology and biochemistry from the Queen's University in 1995. He subsequently served as a postdoctoral fellow at the University of Calgary before joining the University of Manitoba in 2000. At the University of Manitoba, he has held academic positions as assistant professor, associate professor, and since 2013 as a professor. Throughout his career, Professor Ivan has demonstrated exceptional leadership in both academia and broader scientific community, serving as Associate Dean of Research and Graduate Studies in the Faculty of Science, Manitoba University. He has also held major leadership roles within the Canadian Society of Microbiologists and has contributed extensively to national and international scientific advisory and conference committees. Professor Ivan is also widely recognized for his editorial contributions to many leading journals in microbiology and plant sciences, including service as associate editor and editorial board member for multiple high-impact publications in the past, as well as ongoing editorial leadership roles in the Canadian Journal of Microbiology, Frontiers in Plant Science, and other international journals. His research program has made significant contributions to the field of plant-microbe interactions, particularly in the biology of nitrogen-fixing symbiosis between rhizobia and legumes. His work spans microbial physiology, genome biology, nutrient metabolism, and ecological competition in agricultural systems, with a strong emphasis on improving sustainable agriculture through microbial innovation. Professor Ivan has authored an extensive body of influential research publications in leading journals such as Molecular Plant Microbe Interactions, Microbiology, Frontiers in Plant Science, and Applied and Environmental Microbiology. In addition to scholarly output, Professor Ivan has secured substantial competitive research funding, including major grants from Genome Canada and Natural Science and Engineering Research Council of Canada, supporting large-scale research programs focused on bioinoculants, crop resilience, and microbial ecology in agriculture. Today, we are privileged to hear insights from a scientist whose work has significantly advanced our understanding of microbial symbiosis and its applications for sustainable agriculture. Please join me in warmly welcoming Professor Ivan to deliver the plenary address. Thank you. The podium is over to you.
Thank you, madam, for your nice introduction. It's our honor to invite Professor Ivan.
Can you put which one is it? Thank you. Okay. Is my voice carrying okay? Okay. First of all, thank you very much for inviting me. I am incredibly humbled at everything that's been said here, and that to be here, it's incredible. And I've been very overwhelmed with the country in that I've been here for about a week, and I have I've been shown many places. I've talked about 4 times so far. And today what I'm going to go through is use of biological inoculant in agriculture. Okay, I really reflected on the title or the theme of the conference, which was Smart Solution to Agriculture. And usually I will go out and talk about my work, and that's really easy. I'm going to sort of give an overview and some of my thoughts. I've also tried to limit how many slides I've put on, because I would really like to state a time limits. And I think I'm going to have a very hard act to follow with all of the traditional dancing and all of the ceremonies. Okay, so what I'm going to be talking to you about is something about we call Project Benefit. And I am one of the co-leads on this particular Genome Canada initiative. It built slowly, and with that, with other foundational grants. And this is one what I would share with my co-lead, Dr. George DiCenzo at Queen's. Okay, let's make sure we go the right way. Okay, so we've heard some of this from the Dean's address, and what we're looking at here is a graph, and it's from our world in data. And so if you look at it, what's shaded in green is what the world's projected population should have been in the absence of the Haber-Bosch process. Now, for those of you who know, and some of you might not, I'm assuming this is a general audience, the Haber-Bosch process was brought about in about 1913. First of all, it was shown chemically that you can make nitrogen, and then it was industrialized. So the tagline for that particular process was literally bred from air. So if you look at the atmosphere that we're breathing, 80% of it approximately is nitrogen gas. It is very, very stable, but yet nitrogen is one of those elements that limits crop or plant growth. What you're looking at in the brown is basically the population as it was projected. So in 2015, approximately 50% of the world's population is only here because of the Haber-Bosch process. So if you think about that for a while, if you look at the person next to you, one of you wouldn't be here. Okay, nitrogen is incredibly essential. The other thing with that I remember as I've been writing some of these grants and looking at things, if we took some of my skin and we made it ash and did an atomic absorption on it, you would probably find that 70% of my nitrogen came out of a factory, and that's the same for just about everybody. So nitrogen has been an incredibly useful, or this process has been very, very useful. It has mitigated a number of famines in the world. And as I go on, we're also going to see that there's a problem with nitrogen. So Project Benefit basically was a call for the mitigation of greenhouse gases in Canadian agriculture. So what I've got on the screen there, if you're looking at it, they're Canadian numbers, but you know, it sort of gives you the breakdown that agriculture does give off greenhouse gases, and most of them as nitrous oxide. Okay, so this morning as I was looking at it, and you're always making last tweaks to your slide, I know that I moved a few slides around, and I thought, oh, this will work better. We'll see if it really does. Okay. I like this particular diagram. It's out of a review that we published more recently, and it's meant to be sort of a general sort of look at it. What we're looking at is a cross-section through a root, and then we're basically looking that in the green we have bacteria about, and there's a few other portions to it. So think how things have changed since I have first learned about plant physiology. First of all, if you look at all the little green parts, those are supposed to represent bacteria. When I took plant physiology and I started learning about cross sections, there were no microbes inside the plant. And when the endophytes were first shown, people used to say, this is an artifact. We now know that, yes, endophytes really exist. The other thing to be really aware of is that plants actively secrete a lot of nutrients. So if we look at a general plant, depending on whose numbers you're going to want to believe, somewhere between 40 to 70% of the plant's photosynthate or nitrogen that they take up is actively secreted or lost through the roots. And somebody might think, why on earth would you do that? You're trying to live. Why would you want to be giving it away? And the secret is, it basically feeds the microbes in and around the bacteria. And so if we go around, and if you look at about the 7 o'clock position on the slide, you can see we have K, P, N, S. Those are going to be elements. All nutrient cycling goes through microbes, okay? Depending on who you are, some people see this very easily, some people do not, but it is one of those where if we're looking at nutrients moving, the microbes need to be considered. So if you want to think of it in human terms, and I often lecture, one of my lecture slots that I have to do is introductory microbiology 1010, so it's to a first year class. And one of the themes that I try to come up with is we live with microbes and we can't get away from that. Everybody, usually in a first year biology sort of stream, wants to become a medical doctor, right? And so you have to sort of tease that out of them and say the environment is very important. So little factoids that you can take home. We have more bacterial cells in and on our body than we have human cells. Okay, now let's extend that to the bacteria. When you are going to have a nutrient source in the soil, that is basically going to harvest bacteria. And so those are going to be colonized, and you're basically going to have a community in and around that. Like what would happen to our physiology, everybody knows the bacteria that are bad, right? From the youngest age, wash your hands. You don't want to, you're going to get sick. But the truth is, like, the bacteria that are bad are really bad. But the majority of bacteria are either absolutely neutral to us, or they have some benefit. In our gut, E. coli, often thought of as a villain, also gives us a vitamin K. That's our source. Now let's go back to the plant. Okay, so now many people can sort of understand their human biology easier. The plant root that's going through the soil, and now you're going to get what's termed the rhizosphere, the area that is going to be dominated and controlled by, be influenced by the plant exudate. That is going to be incredibly important. Okay, a little bit about just a normal look at the farm nitrogen cycle. So we have our little stylized bacteria factory that is making nitrogen. The nitrogen is going to be applied to crops. And then I've got this great big sort of circle about it saying microbes, and we have some of the interaction. So points to remember. When we're looking at this, we notice that as a normal microbial process, nitrous oxide is given off. The way that agronomy is usually worked out is empirically. Every area has its own agronomists. They say, if we put this much nitrogen, this is what the yield is going to be. If we put more, this is what the yield is going to be. And often there's a decision that gets made of how much nitrogen do I put in, and what's my return on my investment as a farmer. What is less known is that up to 50% of all the nitrogen that is put into that system is lost as nitrous oxide gas. Okay, I'm going to sort of switch gears a little, and I'm going to introduce you to some microbes that are called plant growth promoting rhizobacteria. Okay, I was thinking of how to do this, and I was actually sitting in the airport, and I did my own download on NCBI saying, I should be able to do this. I want to know how many publications have plant growth promoting rhizobacteria either in their title or their abstract. In my search, I also found somebody's done it before me, so I said, okay, fine, I like that. Okay, so the earliest reports that I could find of plant, I'm going to just call it PGPR for short, PGPRs is around 1975. I do remember as an early graduate student, as some of the literature started, and you can see that graph starts from about 1996, going to almost the present. You can see that there's more and more people that are talking about PGPR type bacteria. My background really came out of a developed genetic model system. And when PGPRs first came along, it was one of those where you said, there wasn't a lot of in-depth work done. Very much it was, here's something, it works, and then you're looking and going, okay, then another person says, well, here's a different bacteria, and you're going, how does that work? I thought that bacteria did it, not this one. I think the truth is that as we look at this, there's a number of different bacteria that are capable of doing plant growth promoting responses. You can't necessarily look just by species. Sometimes it comes down to a strain level. As in, we can isolate one strain or one species, and one strain will have that effect, another will not. So the point out of this is the evidence is overwhelming. Plant growth promoting rhizobacteria, and some of it we look at just as very early plant growth, but there's other ones where you look at it and they also help with nutrient use efficiency. There's a number of common mechanisms that people have sort of looked at. If we're going around clockwise, obviously We have nitrogen fixation. We have phosphate solubilization is very important. The promotion of some plant hormones, all of these have been shown. But the problem is most of the time, people might look at it and say, oh, look, it makes indole acetic acid. That must be the mechanism for PGPR. And the truth is, very few times have people actually looked and made that direct link. Okay, I'm going to move on from PGPRs and just start looking at something else that is going to all come together. And that is community analysis. When I was a graduate student, I remember being in a lab meeting where the question was, do we sequence this gene or do we do ultrastructure? The argument that carried the day was, what if we sequence the gene and there's no match in GenBank? So that would have been in the mid-'80s. Now that's an impossibility. The other thing to be really aware of is next generation sequencing, as it's come along, has really changed what we can do. It's not solving our problems, but it's allowing us to have a slightly different look. And so what I've got pictured here is what people will call like a core microbiome. And what we've got is plants from 3 different places, and we basically do a 16S community analysis. So for those of you who do not know, 16S is part of the machinery of a cell. It's incredibly conserved. We have an equivalent. Every bacteria has one. Phylogenetically, as people have been looking, how do we look at these things? What they have looked at is said, we'll use this as a comparison. From that, you can look and say, if you have the same plant grown in 3 different locations, and you look to see, as a Venn diagram, what is going to be together, or what do you always find, that's what people will often call as a core microbiome. As people have gone on, now we're going into more transcriptomic studies. It's not what are the bacteria there, but which bacteria are expressing what. And I think those are some of the sort of the foundation of where we're at. So I'm now going to introduce you to what we were very fortunate to get, and that we called it the Benefit Project, which was not what we started with. The actual title for this grant was Bioinoculants for the Promotion of Nutrient Efficiency and Crop Resiliency in Canadian Agriculture. Okay, this slide is one that I look at and have 2 parts to it. We had to keep working through one of the arms of Genome Canada, which is called Genome Prairie, and they were trying to help us get the grant, meaning that in the end, the questions that would, the people that would adjudicate it are going to be external. And so in the background, the overall part is an infographic. And I remember we were told to start writing this grant on December 12th. And it was due toward the end of January. To give you some idea of the scope of this thing, when we finished it, it had 35,000 words to it. It was a tome. It was really a lot. But somewhere in the middle of that, we were told, you need an infographic. And we sort of looked at each other and said, and who's going to do that? So anyways, we did do it, which is one of the reasons we probably got the grant. We jumped through all the hoops we were asked for. And I said, if we get this grant, I'm going to show this as many times as possible. Okay, so this is the overall organization of what Project Benefit actually is. Activity 1 and 2 really focus on the isolation of microbes. Activity 1 is really one where we were looking broadly for basically anything. So the idea was, we know that plant growth promoting rhizobacteria exist. Let's start looking in locations within Canada, within Ontario, Manitoba, Saskatchewan, places where there's a lot of crops that are being grown. And activity 2 was basically all about rhizobium. So both me and my colleague, Dr. Gicenzo, that's what actually drives us. And so within activity 2, the problem that we were addressing was given to us by Manitoba pulse and soybean growers, and that was, they basically said, there isn't an inoculum for dry bean. And we sort of looked, it's a legume, why not? And the reasons for it are probably many. One is that it didn't work very well, so people didn't buy it. The grower groups would basically say, if you want to grow this because it is a profitable crop, basically fertilize it. So if you're fertilizing that crop, go back to 50% of all the nitrogen is going to be lost as nitrous oxide gas. And this project was for the mitigation of greenhouse gases. It also, as we were about halfway through this process of writing the grant, we realized that it's really hard sometimes to get your hands on the microbes. It's incredibly difficult to go from one country to another with a microbe, whereas when I was an early graduate student, you would show up with basically slants in your suitcase and just give them to your friends. I would not go through an airport right now with a slant. I would like to make it to where I'm going as opposed to having to talk to one of the officers. So we decided that we were going to make a strain collection, but with that strain, remember that this is under the guise of a Genome Canada grant. We had the ability to basically sequence everything that we had. Now, overall with that, we understand that not all microbes that we might find are even going to be amenable to industrial processes. How you grow them and then how you release them or put them onto a crop really matter. The other one is, what if we found a really good bacteria, but it takes so long to grow or it doesn't like to grow? And so in activity 3, what we basically have is looking at microbes and asking the question is, can we passage them and adapt them so that they become more industrially amenable? In activity 4 is really where we started looking at the plant microbe activity. If we're doing this for plants, we needed to be able to do it on plants. Within there, we've got everything from field trials to the other part that we were looking at was a lot of the crops that we have been bred. If there are any breeders in this room, I'm going to apologize for what I say next. Breeding works, it really does, and we can't deny that. But by and large, breeders on a whole do not look at what happens in the roots. They put them under very sufficient conditions. They will look at the chlorophyll, they will look at a pod shatter, they will look at disease resistance, all things that are very important. But what they have not done, or often will not do, is look to see what the biological interface is. There are some countries that have done that very well. Brazil, for example, when they started bringing in soybean, their breeding programs completely changed, and what they did was their breeding program brought in biological nitrogen fixation, and that was the end of it. And they've shown incredible results, especially with regards to sustainability. What we were probably looking at is, if we have a good response, and we have the ability to look at it both at 10x microscopy, which allows you to look at spatial expression of genes, as well as we have the ability to look at the single cell sequencing. Can we find the molecular signals for what does a good interaction look like? Why do we want to do that? Well, I'm getting close to the end of my career. I'm not going to pursue that. But the idea is that basically can become a quantitative trait loci that we can move into breeders to basically say, this is something that's good. You should probably screen for these in your germplasms as you go along. With the holistic portion of this whole project, I think with a lot of foresight when Genome Canada came up with this program, is there was the need for the incorporation of social sciences. And so this is what we often call activity 5, where within that we have people in geography departments that are modeling greenhouse gas emission on fields that we're working with. But there's two other parts. We've got an agricultural economist. What he's looking at, and they're looking at, is saying, what is the break point for making any inoculant financially feasible? And at the beginning we thought, okay, if we find something that increases 10%, run it through the numbers, is 10% good enough? Or is 10% just background noise? Or the other way is, how much does it cost to put on an inoculum? What's the return on the investment? Okay, so those are sort of checkbook type exercises, most people get that. The other one that I think is really, really important is we've got down there the demand for the actual product. We could find the best microbe in the world, but if we can't convince a farmer to use it, there's no point in it, okay? And the problem with biologicals, people understand rhizobium because everybody who's growing one crop has a legume in their rotation somewhere. But what is very poorly regulated is these plant growth promoting rhizobacteria. And the problem with it often comes down to efficacy. Will it work? Will it work in my field? So we need to know, or a lot of the questionnaires that we're doing basically looks to see will they adopt it? Or if we end up with a product and it goes on a store shelf and it has a little tag, it says greenhouse gas compliant. Would a consumer pay more for that? And those are open questions. It's just that we need to know this before or as we're moving ahead. Okay. So the deliverables, they're on the last slide, a biobank of plant beneficial microbes, inoculants that could be robust across a number of different soils, as well as the development of microbial products. Another way of looking at this as basically as a Venn diagram. We need an effective product, we need grower interest, and it needs to be financially viable. And so what we're looking at is really that middle ground. Can we find that sweet spot where everything works or comes close to working or is on the road to working, but we've got buy-in from the growers? And okay, so I'm going very quickly through some of the overview. We had an oversight committee review 2 weeks ago, so I stole some of the slides from that. In activity 1, it's basically telling us what we've done. So far, we've annotated genome sequences generated for nearly 2,000 different bacterial isolates, hundreds of species, about 100 genera across 4 different phyla. One of the things that we were very lucky and successful is our buscular mycorrhizals. We have a person on board who has the ability to go from a single spore, and we've been sequencing them, and to date we've got approximately 80 new ones that we have found. This is going to be a massive increase within that pool of people that work on our buscular mycorrhizas. The culture collection that we have, we're getting ready to launch that. We've got it sort of duplicated. There's the open link front page that'll look at it. It is really meant so that researchers can have access to these microbes and that they're going to be around for a long time. If it's an academic, the charges are nominal. It's basically enough to the handling to get it out. Commercially, we're going to ask for more. Activity 2 is all about rhizobium. So this is a slide I've given a few times in the last few days. We basically screened and isolated a number of different rhizobia that are capable of making nitrogen-fixing nodules on dry bean. And basically, this is broken down by species. You can see there's not one species, there's multiple species. Every species has ones that are good and ones that are poor. But within activity 2, we also have a bean breeder. And so with that, what we're doing is we're taking some of our better isolates and we are exposing them to a diversity panel to ask, what is the response? Do we actually have the right backgrounds in the inoculum, into the bean lines that are currently out in production? The activity 3 overview, again, it's very industrial or with the industry in mind. How well can something grow? Can we make it grow better if we passage it? And if we passage it and alternate it between plant and an industrial type growth condition, are we going to lose the benefits that we have? But I've just said that. Activity 4, really, we needed to come up with a way to go through a lot of microbes to see which ones would have plant growth promoting activity. We've screened well over 200, which is more than the milestones that were out for us. We ended up using barley because it was a diploid It also has pretty good genetics with it for when we end up going with this. We are at the point now that within our final year, next summer, we should be putting some of these into the field. Whereas the rhizobia have been in the field last summer, and now we've gone to a proper agronomic sort of trial, 2 sites, two different sites, 2 different seasons. Okay, I'm going to just sort of switch a little bit, and this has been sort of higher up looking down. Here's what something looks like if we're looking at some PGPRs. Very commonly, bacteria that are isolated as being phosphate solubilizing have PGPR traits, and we don't know why. Some people would say, oh, it's because of the phosphate solubilization. It's not always that clear. So in the top panels, you can see what they look like if you put them on a media that has an insoluble phosphate. As they grow, you are basically going to be clearing the media. On the bottom was just basically the quantity to be able to quantitate that. Whoopsie. If you look at what the effect is on soybean, you can see that the isolates that we had, some look very good. One looks really pathetic. But we did these, they had an effect on soybean, but not on wheat or not on canola. So again, you're going to have to look and say, one strain does not necessarily meet all the criteria. We were able to sequence the genomes. I'm pretty sure you can't read these. The ones at the top, I don't know if what the arrow shows at this point in time, were all Paraburkholderia stratomyana. They're interesting because they were only previously isolated in South Africa, and they were isolated as being nitrogen fixing. We also isolated a Paraburkholderia graminis, and the last one on the bottom, which is just a straight up Burkholderia, that was the one that really was not friendly to the particular plant. What we could get out of this was we were fortunate enough that we were able to close our genomes. And when we closed our genomes, this actually, we were able to scaffold the type strain down to it. And we were able to say, you know, we know that it had 2 chromosomes plus an accessory plasmid. Our isolates didn't have nitrogenase, or those are the genes for nitrogenase. But what we did find is it has the ability or the potential, we haven't tested it yet, to grow autotrophically, basically using CO2. Okay, I'm going to come out of this, and the next number of slides are basically going to ask questions. They're going to be some of my thoughts as I was sitting there, and I think it's something we need to think about. I went through and we showed you that I'm very much wanting to know how something works. If I want to know how something works, I want to go down to the gene level, make the mutation and whatnot. Do we really need to know that? And I think the answer is no. And I think that really comes down to, I think there's a disconnect between basic research and applied research. So as I've traveled through the country, I would say that within Sri Lanka, there's a much stronger tie to applied research. I think that that's something a lot of countries need to learn. The other portion is increasingly these projects are getting complicated. I would like to tell you that I understand every part of this benefit project. I don't. I need to sign off quarterly on all the financials, but it really is one where sometimes when you have different fields that are interacting, both may not understand each other, but there's that magic space in between where you look and whatever somebody says, you go, I don't quite get that. Does that mean this? And it might not, but then that's the fertile area that I think that really bringing groups of people together where it's important. We can get a lot of research done, but I think that the important part is we need to keep our eye on, does it work and can we get it to be applied? Okay, are all inoculants fully exploited on legumes? We all know legumes, or many of us know, that legumes have formed root nodules where nitrogen's fixed. But not all legume crops are using inoculant in Canada. Why? In some cases, it is just more convenient to fertilize. They'll say, okay, we're not getting enough return out of this. And I think this is one where I mentioned Brazil earlier. I think these might need to be mostly country-based specific solutions. If we move on to that and look at plant growth promoting rhizobacteria, one of the biggest problems is you can get them to work in a growth chamber. You put them into the field and the results are incredibly mixed. And I think that comes down to there's a scarcity of products, and in places where products are put out very quickly, if they don't work, the farmer's not going to adopt them the next year. And I think that's one of those where we need to be very careful. People using synthetic communities and consortia. In some cases, it can work really well. I've seen consortia of 2, sometimes 3 put together. But okay, I can go through Proceedings of National Academy of Sciences journal, and you find community analysis where they've got 20 to 30 bacteria put together microfluidically, and they've done everything on Earth on it. There is no way on Earth that that's going to work practically. It's hard enough to get 1 or 2 to grow together. So I think it's a strategy that I think that academically, that approach can work to say what might be able to go together. Practically, I think it's people that are doing basic research who are fooling themselves. That comes out of someone saying that if you get a chance to give a talk, never miss the chance to be a little controversial. Okay, is there a need for regulations? This changes from country to country. Rhizobium is fairly well understood. If you go to Australia, they have an arm's length body that basically will look and say, this is the inoculum that we're going to use. It gets a green check. A farmer is not going to buy any inoculum from an inoculant company that does not have that green check off. We don't have that in Canada. It is just basically private companies, here, it'll work. And then you have places where Brazil went through this, and the efficacy for rhizobium inoculation was so poor that Brazil, Uruguay, they all stepped in. And now they're seeing positive results. There needs to be buy-in from all the way across the board. And especially when you have something like a plant growth promoting rhizobacteria. That's much more difficult. And how do you get that efficacy? And I think that when you look at the efficacy, I said Australia has a wonderful system for their inoculants. I talked at their nitrogen fixation conferences, and I was told biologicals are just a mess. People do whatever they want. And I think that that's where we may lose our opportunity, is being able to not only find them, they exist. It's going to be how do you harness them. I noticed when I was doing these slides that they started getting text heavy, so I basically went to Wikipedia or looked and said, give me a result for something about breeding. Breeding works. We have benefited greatly from plant breeders. Increased grain size, increased resistance to common diseases, improved ability to harvest. If you got a pod, will pods shatter? Will they work with the machinery that you're using? But there needs to be a consideration for the microbes. Like, if we look at it, have we lost things from common crops? And I know that there's a study that I read from a colleague of mine in Oxford who went on and they were working with wheat, God bless them, as being a hexaploid. And their conclusion was, yes, some of those lines of backgrounds of wheat do not interact quite as well with microbes as others. And here we are for second to last slide. How do we go forward? And I took this from a review because I really, really liked it. And it's, first of all, we need to build that trust with the farmers, and there needs to be across the entire enterprise. You need to be able to look at this and have buy-in. And the one that I like over here is people will get their soil tested, they'll get the pH tested, they'll get the nutrients. Do I need to amend? Do I not need to amend? One of the ways that could be going forward is if you take that soil, and now something like 16S community analysis is gotten fairly inexpensive, and you can then get a snapshot of what the community might be. And I think that one of the things that we are looking at is trying to use machine learning models to look and say, with all the community analysis that we've got, will this particular organism work on that community? And I think the answer might be if you have a bank of 10 PGPRs and you look at one of these, you'd look and say, these guys are not going to fit in. They just will not work with that community. Whereas when you do the network analysis and look to see what is positively correlated and what's negatively correlated, you might look and say, we got 10, these 5, no chance. Or you could try them if you want, but these ones actually may work. So I think that that's one of the ways that I think it might be a way forward. Okay. The people on the grant who were all the activity leads were all put out front. The people that I've got listed here are people in my work, in my lab, that are directly working on the benefit project. So that would be Dr. Anna Montenenko, Ambahai, who has invited me here. Her work was one of the foundational works that allowed us to do this. Zeel Patel was an undergraduate student who ended up doing a lot of bacterial isolation, and he had the ability to do that. Ishani did an MSc. She came out of the University of Peridiana. She's been a blessing to my lab and some of the practical aspects that she's been able to bring in. Also a pair of hands that works really, really well. Mubashar Bari is from Bangladesh, who the day after the Genome Canada grant was publicly announced, I got an email going, I would like to work on this project. He has since got his MSc. I thought this is borderline creepy. But there's someone who's looking out after himself, and he had a wonderful MSc and going onwards. Within the Department of Agriculture, a collaborator of mine, Kristen McMillan, who does all the dry bean trials for ours, and Dr. George Chenzo, who is the other co-lead on this particular project. And with that, thank you so much for giving me the opportunity to talk. Thank you for your attention. Questions or just leave?
Thank you, Professor, for your engaging and informative plenary address on the use of biological inocula in agriculture. We are truly grateful for the knowledge and the perspectives you have shared with us, and sincerely appreciate the time you have devoted to being with us today.
Ladies and gentlemen, the next important item on our agenda is keynote address. For that, may I kindly request Professor Mrs. Sivamadi Sivachandran to kindly introduce our esteemed keynote speaker, Professor Toshaki Owari, to the audience.
Deans of the faculties, renowned invited speakers, academic, administrative, and academic support staff, distinguished guests, students, ladies and gentlemen. It is a distinct privilege to introduce our visionary keynote speaker for ICDA 2026, Toshiaki Owari, a globally recognized authority in forest science, management, and engineering, whose pioneering work targets the very heart of our global fight against climate change. Professor Owari earned his PhD from University of Tokyo in 2000, and he went to hold distinct academic posts at Hokkaido University and the University of Helsinki, Finland, before returning to his alma mater. Since 2025, he has been served as a full professor and the director of the University of Toyota Hokkaido Forests. For decades, Professor Owari has established the foundational scientific frameworks utilized by forestry scientists, practitioners, policymakers globally to balance economic production with biodiversity conservation. He is a highly prolific researcher with an extensive publication record in top-tier international journals, with an expansive publication record, over 100 scientific papers that have earned more than 1,000 academic citations from researchers worldwide, and also served as the lead editor of the Springer Nature landmark volume, Integrative Forest Management and Silviculture. His extensive bibliography features pioneering papers that have fundamentally advanced digital forestry. For example, improving the estimation of structural parameters of forest using metrics derived from unmanned aerial vehicle, which is published in 2024, and using UOV photogrammetry for estimating stand parameters of plantation using different filtering method in 2023, and recently the spatial location of broad species in mixed forest using UA multispectral spatial localization of broad species using multispectral imagery and mask RC doublet models, which was a recent publication published in 2025. Beyond his research, he is a true pillar of the global academic community, serving as the deputy coordinator for the silviculture division of the International Union of Forest Research Organization. Professor Owari bridged countries' all forest wisdom with cutting-edge spatial computing by integrating airborne LIDAR, drone photogrammetry, and graph convolutional neural network. His methodologies allow us to map complex ecosystem, track long-term carbon stocks, and model biodiversity with highly precision. His pioneering technological framework is profoundly related to our work in Sri Lanka as we look to protect our own unique regional ecosystems. Furthermore, I take this opportunity to express our institution's deepest gratitude to you, Professor, for personally mentoring our own staff, Dr. Javanan, with the expertise to utilize the advanced digital drone technology and the spatial mapping tools, which is a new area for Sri Lanka. He has been inspired with a new direction for our future field research and let us invaluable asset to our institution. Ladies and gentlemen, to guide us into next frontier of digital ecology, please bring your hands together to our visionary keynote speaker, Professor Toshiaki Owari. Professor, we welcome you to the podium to present your address on digital transformation in forestry, challenges and opportunities. Over to you, sir.
Thank you, Professor, for your kind introduction. Vanakkam. Hello. It's my great honor to be here and to share our experiences with you, University of Jaffna, Sri Lanka. And I would like to thank, especially for inviting me, especially to conference organizer, Dean, Professor K. Pakera-san. Sorry, it's difficult to remember. And one of the joint secretary, Dr. K. Jayabanan, who was my previous PhD student. And thanks for invitation. And I'm now enjoying visiting Sri Lanka. This is first time for me here. It's a beautiful country. Food is very nice, and people are very friendly. I like it. Thank you very much. Okay, so my talk is about digital transformation in forestry. And the topic of this conference is smart solutions for commercial agriculture. So it somehow fits the topic, and I hope you will have some ideas for your future efforts, research. Okay, so I will cover the following 4 topics. Let's go through the introduction. Have you heard the word digital transformation or DX? According to the business literature, DX is defined as a change in how a firm employs digital technologies to develop a new digital business model that helps to create and appropriate more value for the firm. In Japan's forestry sector, forestry DX has progressed rapidly, and it is defined by the Japanese Forestry Agency as initiatives that utilize technologies such as remote sensing, AI, and ICT to fundamentally transform conventional forestry operations through data digitization and utilization. In Japan, DX addresses low labor productivity and high occupational risks, while simultaneously enhances the attractiveness and sustainability of the forestry sector. And the Japanese government aims for full utilization of digital technologies by motivated forestry managers by 2028. In northern Japan, the University of Tokyo Hokkaido Forest, UTHF, we manage uneven-aged mixed conifer broad-leaved forests under the stand-based silviculture management system, or SSMS. And we have actively introduced geospatial information technologies and digital tools since the late 2000s. As a representative case of forestry DX in Japan, my talk presents recent technological developments at UTHF and discusses how digital technologies can contribute to the development of forest management systems. Okay, so let's move on to the second topic. I would like to introduce something about our university forest. In University of Tokyo, we have a total of 7 branch forests in Japan. And our forest is located in the northern part. So, and it was established in 1899. We have more than 120 years history. And the area stands at 22,000 hectares. And we have adopted the standard-based silviculture management system since 1958 to implement its unique close-to-nature forest management and planning. So we have nearly 7 years experience for this forest management. The concept of SSMS, or Rinbun Seigyoho in Japanese, the management should be adaptive to the conditions of each stand for maximizing multiple public and economic functions of the forest. And we have 6 basic principles: promoting and maintaining pre-climax stand condition, avoiding intensive forestry operations, adapting management to the specific conditions, maintaining tall, multi-layered stand structures, retaining trees with superior traits, and promoting mixed conifer broadleaf forest composition. So we normally do not do clear cutting, but single tree selection cuttings are main harvest way of harvesting. So if you look at the graph on the left-hand side, you can see the x-axis is the year and y-axis is growing stock per hectare. So you see the blue line, which shows the stand development of managed stand under single tree selection system. So we cut partially and grow, partially grow. So this is a typical trend of development. So you can see the picture on the right-hand side. So it looks like a natural forest, a managed forest. We have beautiful mixed forest, so evergreen conifers and deciduous broadleaves. But we have been managing this stand every 10 years or 15 years, and in 50 years, we have done harvesting for 5 times. But still, we can maintain this kind of stand structure. The work procedures under our silviculture management system. So we first do forest type classification and forest inventory to plan allowable cut, and single tree marking for harvest, stumpage sales and logging operations, and scarification planning on poorly regenerated site. And I will explain more about our management later. And as the professor kindly introduced, we have just published a new open access book from Springer. The title is Integrative Forest Management and Silviculture, Harmonizing Conservation and Production. So you are welcome to visit this site. And so this is open access, so it's free for reading. Okay, so this is the main topic, and let's move on to the third topic, forestry DX in practice. So you can see drone. It's currently common practices for us. So first, I will introduce about how we use digital technologies for our management and especially forest type classification. So if you have the opportunity to visit us, so in the northern part of Japan, Hokkaido, you can observe our natural forest. But stand structure is not uniform. So we have conifer dominant forest, broadleaf dominant, or stand density is different, so you cannot manage this type of complex forest as a constant or uniform way. So that's why first, one of the essential techniques for us is to classify our forest into several stand type or forest type. So if you look at this map, on the left-hand side, you can see the aerial photos. So this is one of the managed site for us, maybe around 400 hectares or 500 hectares. And you can see on the right-hand side a map. This is our forest type classification map. And each color shows the forest type. And we mainly focus on the forest area with green color, which is we have sufficient number of conifers and good natural regeneration. Even if you can do a single tree selection cutting, you can expect to recover the growing stock. But for example, it's difficult to see, but we have light blue color area, which is what we call sparse forest. So canopy gap, few number of trees exists. So if you do cutting, we will lose the quality of stand. So that's why if we manage these stands, we should do some scarification and planting work to assist regeneration. So this is quite important silviculture planning technique, but also it is very laborious, hard working. Until 2009, we use this kind of manual surveying to delineate the boundary of forest type. You see, so distance is measured by rope. But after 2008, we started to use digital devices. This is a laser range finder with electronic compass. So now you don't have to use a measuring rope. Then 2012, we started use GNSS surveying. So are you familiar with the word GNSS? It's a global navigation satellite systems. So you can obtain the exact location within the forest. So based on the satellite signals. And currently, we are using low-cost RTK GNSS receivers, which is much more accurate even under canopy. So sub-meter positioning accuracy is possible. And the price is fairly okay comparing to the past. It was very expensive, but now it's okay. RTK means real-time kinematic. So we receive the satellite collection dataset to increase the positional accuracy. We have done airborne LiDAR surveying, so laser scanning from the sky for whole area of our university forest. And we can get very accurate and precise digital terrain model. So this is a resolution of 15 centimeter grid. And we also have a digital surface model by subscribing DSM from DTM. You can calculate this digital canopy height model. So you know the height of canopies of whole area of the University of Forest. So this is a short video about the use of fixed wing UAV. We used this digital device between 2015 and 19. So this is, but we don't use it anymore. So this is quite expensive, but it's a little bit, you need more space for departing and landing. So it was a little bit difficult to operate. So, but we published based on the data set pictures taken from the UAV, and by using softwares having the function of structure from motion technique. So stereo images, you can calculate, you can reconstruct the 3D model of the forest. This graph shows, so red dot shows the point cloud taken from airborne radar data, so laser scanning, and blue dot shows the canopy height information reconstructed from SMF software. So the accuracy is quite good, although it has some limitations. So now you don't have to request companies to fly and scan for forest, which is very, very expensive, but we can do it by ourselves. So it is very flexible, and we can reduce the cost for the data collection. So now, currently, we can easily make this kind of orthomosaic photos over the managed site, and also we can easily generate canopy height model. So this is a canopy height model created from airborne radar dataset, but also so by creating UAV images. So it is very similar, so which means quite accurate. And by using this data set, we can make a statistical model to estimate merchantable volume of the forest and also carbon stock. So this is just an example done by one of my PhD students. Oops. So in forestry, we typically use a plot-based survey, but now we can estimate the whole area, the spatial distribution of forest resources. Since 2014, we start using multi-rotor UAV, so drone, drone. And after 2022, we started use multi-rotor RTK UAV. We used to need, we needed to have a ground control point at the same time with UAV flight, but now we don't have to set the ground control point anymore, because this is very accurate based on the RTK technique. And we also use different type of sensors, not just RGB photos, but also multispectral UAV. And so I have heard from Dr. Jaffna that so this university already introduced this DJI multispectral UAV. So this is very effective. You can calculate based on the near-infrared band, so you can calculate different type of vegetation indices. And you see this is a forest, but you can see the canopy gaps. But if you take photos by multispectral UAV and calculate the vegetation indices like NDVI value, so you can easily specify the location of canopy gap. Or you can use this kind of information to identify a tree species or another kind of more accurate information. Currently, we are now trying to use another sensors, so laser scanner sensor mounted on UAV. So now you can easily create a 3D model of your forest if you use these devices. One of my PhD students, Nyomi Hum, conducted broad species identification using multi-spectral UAV images. And she compared how accurate is it. And this is based on the deep learning technique. So you see the upper side and right-hand side, it is answer or annotated tree crown. And bottom pictures are result of the data analysis. So pick up the tree canopies and try to do the species identification. And if you use both RGB and near-infrared data set, and also a canopy height model, it's a fusion of the data set. You can get more accurate broad species identification. This is the result of the work by your faculty member, Dr. Jaffna. He conducted tree species classification at our university forest. And he used two types of machine learning technique and also UAV images. Left-hand side is RF, means random forest estimation, and right-hand side is deep forest convolutional neural network species map. So right-hand side gives a more accurate tree species classification. But it's a very good trial. You can see the picture of Dr. Jaffna. Okay, so let's move on to another digital devices for forest inventory work. We currently use tablet devices. We do not anymore use the paper to describe the data for measuring, but we use a tablet terminal like iPad or something. And currently we are trying to introduce the handheld LiDAR or backpack LiDAR. So you see a laser scanner carried in the back. Or this is another type of terrestrial backpack LiDAR scanner named MAPRI sensor. This is relatively low cost compared to other devices. And we are now trying to implement this and for plot-based inventory work. This is a part of our international joint research project with Slovenia, which is one of the European countries, so former Yugoslavia, eastern part of Europe. Slovenia has a long history of close-to-nature forestry, And we have some experience for the use of digital technology. So it's a good collaboration for each other. It was a 3 years research project. And we brought our digital devices to Slovenia and conducted forest inventory and compare with the conventional way of data collection. Okay, so next part of procedure is a single tree marking for harvest. We have a total of 25 technical staff in our university forest, and so they go to forest and they decide which tree should be cut for harvest. And our tree marking guidelines shows that we do not select expensive or high quality tree. We try to remain, but we select from the decayed tree or dying tree or trees which suppress the young trees. Then forest stand can reactivate and start grow more. Since 2008, we used this kind of GNSS receiver for tree marking practices. So we mark every tree in the forest. And currently, you don't have to push button for obtaining the position. But if you input the data about the marked tree, it automatically records. The position is automatically recorded by synchronizing the time of tablet terminal and time of GNSS receivers. So she just select what kind of species, what the size, the quality, then just one button. Then you can automatically get the locational data also. So this is our management planning map, forest type classification created by ArcGIS. And all marked trees are recorded like this. So 1 year, 20,000 or 30,000 trees are collected. So this is useful, practically useful to sell trees as a stamp page, and it's a good data set for scientific purpose. So we know anthropogenic disturbances made by selection harvesting. So we can accumulate the history of our management on a precise way. Currently, we use new GNSS receivers, so this is very small, and you can get more accurate locational data. So the locational data was not so accurate used to be, but now, using this kind of devices, so the accuracy is much now higher and not so expensive. Okay, and briefly, I'd like to introduce this logging operation, the use of digital technologies for the logging operations. So we use machines to collect logs from the forest. This is a forwarder. So grapple loader put logs behind and just go to the roadside. And you see that we put moving-based GNSS receivers. So there are 2 antennas, and you can obtain not just the location, but also the direction of the machine. So we can use this kind of machines to check the productivity, operational efficiency, and to prevent forest damage caused by this kind of machine moving. So we can accurately plan the skid trail and so try to reduce the number of damaged trees. And also we have to do some scarification work or assisted regeneration work, because we have very thick understory vegetation, sasa bamboo, so it impedes the germination or growth of seedlings. So we need to remove sasa bamboo by machine, and you need to plant mainly conifer seedlings, because you cannot expect natural regeneration. So this kind of silviculture promotion work is one of the now main challenges for us, because in Japan the population has been decreasing. It's difficult to get enough number of workers, so we need to do more efficient operation. And this is one of our recent trial using heavy lift delivery UAV, not just taking photos or not just taking LiDAR data, but lift the logs and extract from the forest. It's a very big stuff. But it is one of the promising technique for us for future silviculture operational work. So we try to use this type of UAV for carrying seedlings, container seedlings in the forest. Okay, so the last topic, challenges and opportunities. Forestry DX offers significant opportunities, but successful implementation requires overcoming financial, human resource, and institutional challenges. So I try to summarize 3 parts, challenges and opportunities: finance and economy, capacity building, and new management models. Finance and economy challenges: high initial investment you need to pay initially, and maintenance software costs, data storage and management, staff training expenses, and limited financial resources. But we also have opportunities: improved operational efficiency, reduced labor requirements and harvesting damage, better management decisions, long-term economic sustainability, government support for DX devices or something, carbon neutral and nature positive funding. So forestry DX requires upfront investment, but can generate long-term economic value. Capacity building challenges: need for new skills like remote sensing, UAV manipulating, data analysis, unlimited training opportunities, aging workforce, resistance to new technologies. But opportunities: safer operations, less physically demanding work, greater attractiveness to younger generations, university, industry, government collaborations, and emerging career. So now in Japan, so some people can start a new business for this kind of smart forestry. People as important as technology in digital forestry DX. So in our university, we have field courses, and we invite students to our university forest and show the latest digital technologies. So they now know what to do, what it is, and how to do it. And we also invite local foresters in Hokkaido, like a state forest, a prefecture forest, a private forest, and we organize a training seminar to show how practically use our digital technique. And lastly, new management model challenges, organization resistance, so all the people don't want to use new technologies, It's a very complex data integration complexity, continuous monitoring and requirements, data standardization, sharing, long-term system reliability. But by integrating UAV, LiDAR, GNSS, AI, and cloud platforms, it enables real-time forest information, site-specific management, improved efficiency, transparent decision-making, carbon management, biodiversity conservation, ecosystem service assessment. DX enables a shift from experience-based to digital data-driven forest management. So at our university forest, we are trying to develop a new management model based on this kind of digital technology and spatial data set. So by using spatial data combined with conventional inventory work, we can accurately estimate the forest resources and which can be used for silvicultural planning and to evaluate ecosystem services. Okay, so in conclusion, forestry DX has the potential to support more resilient, sustainable, and data-driven forest management. Successful DX requires long-term investment in human resources, institutional collaboration, and adaptive management systems. And integrating digital technologies with silvicultural knowledge and field-based management practices is essential for sustainable forestry. So there is one take-home message. Forestry DX is not just a technical transformation, but also transformation of people, organizations, and forest management itself. Thank you very much for listening. Thank you. Nandri. Oops.
Thank you, Professor, for your inspiring keynote address on digital transformation in forestry, challenges and opportunities. We greatly appreciate the valuable insights and perspectives that you have shared with us today. Once again, we sincerely thank you for sharing your expertise and inspiring us with your knowledge and experience.
Ladies and gentlemen, we are truly grateful to our distinguished speakers for sharing their valuable insight with us during this session.
Now, it's a time to honor our invitees. We kindly invite the conference chair, Professor K. Bahirathan, to present the memento as a token of appreciation to our esteemed invitees. May I now invite our plenary speaker, Professor Ivan Orsnik, to receive the token of appreciation. Next, I kindly invite our keynote speaker, Professor Toshiaki Owari, to receive the token of appreciation.
We kindly invite the conference convener of the conference, Dr. S. Tadshanitz Khan, to present the memento as a token of appreciation to the conference chair and Dean, Faculty of Agriculture, Professor K. Pakirathan. Thank you, sir, for presenting the token of appreciations. Please take your seat.
Thank you once again to our plenary speaker, Professor Ivan, and our keynote speaker, Professor Toshiaki Owari, for enriching this conference with your expertise, valuable insights, and inspiring presentation. Once again, a special note of gratitude goes to our sponsors whose partnership has strengthened the ICDA 2026. Our platinum sponsor, Jacob Presida, University of Jaffna. Gold sponsor, National Science Foundation, Sri Lanka. Bronze sponsors, Avon Pharma Chem Private Limited and World Vision. Other sponsors, Mr. Ponnambalam Suryakumar from USA, Northern Modern Irrigation, and Hatton National Bank. We extend our sincere thanks to each of them for their valuable support.
Ladies and gentlemen, as we approach the conclusion of this inaugural session, it's time to express our heartfelt gratitude to all those who made this event a success. To deliver the vote of thanks, I now invite Dr. Mrs. Ambihai Sayanthan, Joint Secretary of ICDA 2026. Please join me in welcoming her to the podium.
Good morning, all. This is my privilege to acknowledge all those who have contributed to this ICDA 2026. So the organizing committee is proud to host 12th International Conference on Rhizome Agriculture successfully. As one of the Joint Secretary, it is my privilege to deliver the vote of thanks on behalf of the organizing committee. First and foremost, I wish to express my sincere thanks to our Vice Chancellor, Senior Professor Trinavukarasu, Trinavukarasu Velnambi, unfortunately, who is not able to join with us physically today, but who gave granting the permission to continue all this activity. And also, we would like to take this opportunity to express our sincere appreciation to our esteemed plenary speaker, Professor Ivan Oresnik from University of Manitoba, Canada, for accepting our invitation and graciously sharing his valuable insight and expertise in the field of microbiology. Our sincere thanks extend to our esteemed keynote speaker, Professor Toshiaki Owari from the University of Tokyo, Japan, for accepting our invitation and for his valuable contribution through the insightful keynote address titled Digital Transformation in Forestry Challenges and Opportunities. We would like to extend our sincere appreciation to all the invited guests from administrative services and agriculture sector for accepting our invitation and gracing this occasion with your presence. Your valuable presence here today is a great honor for us and adds significance to these events. To this session shares Emeritus Professor C. Sivajoganathan, Professor T. Mitsunaka, Expert Jacob Presida, Dr. Y. Sasaki, Jacob Presida, Professor Adhikari, Rajarata University of Jaffna, Senior Professor G. Thirukumaran, Professor S. Vasandarova, Professor Mrs. Sivamadi Sivachandran, University of Jaffna, those who are going to be serving as the chairperson for the upcoming sessions. Thank you for your valuable service in guiding for the upcoming session. We also extend our sincere appreciation to the evaluators for the Best Presentation Awards for your valuable time, expertise, and commitment in assessing the presentation and recognizing the outstanding performance. A very special thanks to all the reviewers who dedicated their time to critically evaluate the abstracts and papers, helping to enhance the quality of the academic content presented at this conference. To all the presenters, authors, and participants from various institutions and regions, we extend our sincere appreciation for your valuable contribution and active engagement. Your participation and enthusiasm have greatly enriched this conference and contributed making this ICDA 2026 a meaningful and successful event. We extend our deepest appreciation to our esteemed sponsors, JICA Presida, National Science Foundation, Avon Pharmaceutical Private Limited, World Vision, Mr. Ponnambalam Suryakumar, USA, and Northern Modern Irrigation, and Qatar National Bank for their generous financial contributions and valuable support. Your contributions have played a significant role in making ICDA 2026 a success, and we sincerely look forward to your continued partnership and sponsorship in our upcoming years. Our heartfelt thanks goes to Professor K. Bahiradan, Dean of the Faculty of Agriculture, as well as the heads of the departments, Assistant Registrar, academic and non-academic staff, and students of the Faculty of Agriculture, University of Jaffna, for their dedicated efforts and continuous support throughout the organizing event. A special thanks must be given to Professor S. Chivachandran and her team and students for beautifully decorating this conference hall and creating such an inviting and eye-catching atmosphere. We are also grateful to the Deputy Registrar, Work Engineer, Administrative Staff of the Kilnoki premises for their invaluable logistic arrangement. To our valued media personnel, we extend our sincere appreciation for your coverage and support in bringing ICDA 2026 into the public spotlight. Your effort in sharing the highlights of this event with the wider community are greatly appreciated. Finally, we extend our warmest thanks to all the distinguished invitees who honored us with their presence today. We sincerely appreciate your participation and look forward to welcoming you to today's afternoon session. In conclusion, I would like to express my heartfelt gratitude to each and every individual who made this event successful in many ways to the success of this ICDA 2026. Your valuable support, dedication, and commitment have been instrumental in making this event successful. And also, if I miss anyone, I wish to thank all if I have missed, and please accept our thanks. Thank you all.
Thank you, Dr. Mrs. Ambige Sayandan, for your words of gratitude. With that, we conclude the inaugural session of 12th International Conference on Rice on Agriculture 2026.
Ladies and gentlemen, we would like to remind you that the conference will continue with parallel technical sessions A, B, and C, scheduled from 11:35 a.m. to 12:35 p.m. Detailed information regarding the sessions and venue are available in the conference program schedule.
Now, we will proceed with the group photo session. For that, we kindly invite speakers, delegates, deans, academics, presenters, and all other participants to come forward and take your positions to take the group photo.